Hi everyone, thank you so much for joining us today. My name is Nicole and I'm a master's candidate in the Sheardown Lab. As part of my job doing research in the Sheardown Lab, I work with living systems like cells, living cells. Cells, if you've never heard of them before, are kind of like little bubbles that make up everything that we are. They make up our tissues and our organs and by virtue, us. But they couldn't do any of that. They wouldn't know where to begin at all if they didn't have some sort of secret recipe or code to follow. In many plants and animals, this secret code is made up of a very special molecule called DNA. DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. It's pretty long, kind of a tongue twister, so we just like to shorten it to DNA. And it kind of looks like a winding staircase. The structure of DNA, if you can imagine it, is like a winding staircase in a castle that spins on itself. It's like a helix. Each step in that staircase is represented by two letters that are paired together, and the railings of that staircase are the phosphate backbone, which is something that keeps DNA in its shape. It helps it hold its shape. The pairings of these letters and the organization of these letters read off like a secret code that your cells will read, and that will tell them what proteins to make, when to make them, and that helps determine everything from our eye color, our hair color, how tall we are, and even whether or not we can roll our tongues. That's all DNA. DNA's letterings, or the letter pairings, are made up of only 4 letters. So there's only 4 letters that make up all of DNA in our cells, in animal cells, and in plant cells. These 4 letters are bases, another type of molecule, and they are adenine, A, thymine, T, cytosine, C, and guanine, G. The organization of these letters is what makes up the secret code. It's what makes the recipe. So if you think of DNA kind of like a recipe book, there's a bunch of different recipes in your recipe book that tell you how to make things like muffins and cakes and everything like that. Each recipe is unique and is specific to one thing. That's how DNA is kind of organized too. DNA is organized in genes. The genes are like your specific recipes. So you have a gene that represents something associated with eye color, and you have a gene that's associated with your skin tone and things like that. Each gene is unique to each person, and it's unique to the organism that it is a part of. So today what we're going to be doing is we're going to be making either necklaces, bracelets, or keychains. It all depends what you want to make, that are based off of different genes that researchers have found. There's entire fields of study. That means there's whole groups of people, of researchers, scientists, that look specifically at genes and what they code for, what proteins they make up, how they're organized in different animals, what happens when certain genes aren't functioning the way we expect them to. And because these researchers do this work, we learn a lot about how our bodies fight infection, how we're related to each other, and how we're related to different animals. It's super interesting work, and it also is what allows us to look at these sequences that we have here today. So in the previous post on this page, there was a handout that has some genetic codes that we're going to be looking at. It also will be posted below in the comments in case you can't find it. So each of these patterns are parts of gene sequences or DNA sequences from different animals. So we have chimpanzee, trout, human, butterflies. We even have some really cool like plants like sweet oranges, carnivorous plants, corpse flowers, things like that. So you're just going to choose whichever pattern or whichever gene, whichever animal that you're interested in, and we are going to be making our bracelets. Now you can make the bracelets, all you need is some string and beads, but if you don't have that, that's totally okay. You can also make the beads out of paper that you've colored and punched holes into. You can make the patterns out of clay and avoid making a bracelet, just make it out of clay or color it out. I myself don't have any beads, so instead I'm going to be using some Fruit Loops that I've organized. And yeah, just get creative with it. Whatever works. It's not that serious. We're just gonna work with what we have. So what I've done is organized some Fruit Loops ahead of time, and I've assigned them to be each of those bases that we were talking about, so each of those letters. So T is going to be orange Fruit Loops, A is purple, C is pink, and G is yellow. It doesn't matter what colors you can use whatever you like. These are just the colors that I decided to use. And I've just sorted them beforehand and labeled them so it's easier when I look at my pattern from my sheet and start stringing beads. So I think I am going to do the chimpanzee sequence because chimpanzees are one of my favorite animals. They're actually the most closely related animals to humans, and there was a researcher named Jane Goodall who studied chimpanzees, and she was actually the reason I decided to go into science. She was somebody who inspired me a lot. So I'm going to do the chimpanzee sequence, and to start, all you're going to need is a string. So I'm using yarn, about 30 centimeters long. It could be longer, it depends if you want to wear it as a necklace or a bracelet, or if you want to tie a loop at the end, just like that. You can use that as a keychain and just loop it through on your backpack or on your water bottle. So we're going to start with the sequence. So I have my string, and I'm just going to tie, you can either tie a knot at the end or you can tie a bead at the end. So I tied the first base that it appears in my chimpanzee sequence. So the first base is G. So I took a G bead and I've just tied it to the end there like that. I only did this because the holes in the Fruit Loops are pretty big and I didn't want to tie too many knots and prevent it from falling all off. So I just tied a bead at the end. So my first bead is G and I have it tied on there. And my second bead is T, or my second base. So I'm going to get a T bead. I'm just tying a knot at the end of my string too, so it can keep the string holding its shape. So I'm just going to take a T bead, which is orange, and string that through. Like that. So the next base is A for adenine, so I'm going to put an adenine bead. So I have G, T, A, and then the next three bases on my sheet are T's. So thymine. So I'll just string that through. And you just keep going until you finish your whole sequence there. The most important thing is just being consistent. So if you made thymine or T, whatever color you made it, anytime you see T, make sure you're using the same color. And same goes for A, C, and G. So just keep going, following the pattern, and eventually you'll be finished. So I actually have one already that I made right here. So this is my chimpanzee sequence. I've tied a loop at the end. So that's kind of what I mean by making a keychain. You can tie a loop at the end like that. You also could use the loop just to tie it around as a bracelet. I probably won't use this as a keychain because it's food, so I might eat it later. Or you could just use the sequence as is and have like a longer string like that and just tie it around as a necklace, whatever you want. So here's my completed sequence. I just followed the letter pattern that was on my sheet, and I used the colors that I set to be each letter, and I was consistent all the way through. So DNA, if we think back to what I was describing before, it's that spiraling staircase. And the reason it looks like that is because it's actually made up of two parts. You have one sequence on one side, and then next to it, you have a complementary strand or complementary sequence. What this means is you basically have the same sequence pattern, but made up of a complementary base. Kind of weird and confusing. Basically, anytime you see adenine or A on one strand, across from it on the complementary strand, you're going to see its best friend. And its best friend base is thymine, T. So A and T always like to be across from each other. They're paired up together. So if you see A on one strand, the complementary strand would have T on it. Same goes for C and G. C and G are best friends. So if you ever see G on one strand, on the other side of the DNA sequence, or on the other side of that staircase, you would see C. They always like to be next to each other. So if you've made your sequence already, just one side of it, you can keep it like this. This is great. I have a bunch of single sequences here. So these are actually ones that I was gifted when I did this activity with campers. So this is human DNA, and it's just a single strand. And it looks great like that. I like it like this too. It's super fun. But if you want to try something different, you can choose to also do the complementary sequence. So it's super easy once you get the hang of it. To do the complementary sequence, all we're going to do is match up what we already have in our strand with its best friend. So why don't we try that? So here I have two strings that I already tied together. So if you're going to do both sides, you can just tie two strings together and do one sequence, the sequence from the sheet, on one of the strands, and on your other strand of string, you do the complementary sequence. Or if you did one and now you want to try to do another strand, just start stringing another strand, the complementary strand, and then tie them together later. Whatever works. So I have my other strand here, it's already tied, and I'm going to use the pattern that already exists here for me to do the complementary strand. So if I look at my sheet and I look at my pattern, my first bead is yellow, which is G. I made G yellow. So G is my first base on this strand. That means on the complementary strand, it has to be G's best friend. So G always likes C. So if I see G here, I'm going to string C on my complementary strand. So I have a C bead here, it's pink. I'm going to string that on. Now the next base in our sequence is T. T's best friend is A. So anytime I see T on that strand, I'm going to put A on the complementary strand. Like that. The next base is A. So A pairs with T, so I'm going to put T instead, because that's its best friend, that's its opposite. Then we have 3 T's in our original strand. I can see that on my sheet, so I'm going to put 3 A's. Like that. And keep going. So everything's an opposite. If I see G, like the next bead is G, I'm going to put C. That's my C bead. And you just keep going, putting the best friend next to it, all the way until you get your second strand finish. So I'm about a quarter way there. I think I'm going to stop there. But you just keep going until the end. And as you can see, they kind of match up. Just make sure it's at the end there. So this was our original strand, and then this is our complementary strand. So we start with G, G pairs with C, then we have T, T pairs with A pairs with T, T's all here, they pair with all A's, and then again, G pairs with C. So we just keep going. You have the same sort of pattern, but it's just the opposite. In DNA, at the end, once you get your whole strand, if you twist it, like that, oh, it's kind of hard because I'm not completely done, but if you were to twist it and it wraps around itself, that's kind of the shape DNA has. It's called a double helix because it has 2 strands and it's twisted. And that's what DNA looks like. In our cells, it's twisted in a double helix and then super coiled, like squished super, super tight. And that allows about 2.5 meters of DNA. If you were to take it out of one cell and stretch it out, 2.5 meters can fit in one microscopic cell, which is pretty cool, but also pretty important. You need a lot of information to make up everything that you are, and it needs to fit in a tight space. So once you're done, you can tie it and wear it as a bracelet, or you can wear it as a necklace, or you could loop it, as I've done here. So these are the ones that I've made previously with some beads, and I loop it and put them on my backpack. Like I was saying before, each of these sequences represents parts of genes for those animals. So I did the chimpanzee, and the chimpanzee gene that we coded for, it's also in your worksheet if you want to learn more, codes for a special protein called granulysin. This protein is a toxin that immune cells in the chimpanzee's body will make when the chimpanzee gets an infection and is sick. So if a bacteria or a different type of pathogen, something that's a foreign body that can make the chimpanzee sick, enters its bloodstream, then these special cells in their immune system will make granulysin, and they'll read this genetic sequence to make the granulysin so they can fight that infection off. Pretty cool. If you made the human sequence, you actually made a gene that is called the OCA2 or the OCA2 gene. And this gene is responsible for your eye color. If it is coded for, which means if your cell reads that gene, it will allow you to make pigment to make your eyes the color brown. But if it's silenced or turned on or off in certain ways, then you get different eye colors like blue and green, hazel, things like that. So once you're done, if you did your single strand or you tried the double strand, you can try to do more of the sequences that are on the sheet, or you can change it up and either change the color of beads or introduce something called mutations. Mutations are changes in a genetic code that can happen when bases, so when the beads are either deleted, so you have bases removed, and that could be one or a whole chunk removed. It can also happen when they're switched out. So say your sequence is supposed to have an A in there, but instead of the A in its place, there's a T or a C or a G, and they're switched. A mutation can also happen when parts of a sequence are completely flipped around, so they're in the opposite direction than you'd expect them to be. Mutations are super neat because they introduce variability in our DNA. They allow us to either make proteins that don't work exactly the way we expect them to, or sometimes they actually don't have an effect at all because it's kind of like when you misspell a word and one of the letters is different, but you could still read what the word is supposed to say. Mutations also can allow us to have different features, like 2 different color eyes. Instead of having 2 brown eyes, I might have a brown and a blue eye if I have a mutation that allows me to do that. So they're super neat. If you're interested, you could introduce mutations into your sequences and decide what that mutation would do in that protein or in that animal. You also can use the 4 bases and make up your own genetic code to some of your favorite like mythical creatures, like if you want to make a unicorn DNA strand or a fire-breathing dragon, and decide what that protein codes for, like if it's part of the DNA that helps with the dragon's flying or wings or something like that. So it's totally up to you. You could do whatever you feel like. If you're interested, I encourage you to learn more and research more about DNA and different sequences and things like that. But definitely try to play around and learn about the sequences we have here first. And that's it. All you need is the 4 letters. Just make sure you're consistent. And thank you so much for joining us. Thank you for exploring one of my favorite molecules with me, DNA. And I hope to see you all soon. If you have any questions, please feel free to leave them below and one of us in the Sheardown Lab will get to you. And I'd love to see what you all came up with. So if you have a bracelet or a keychain or something that you did, feel free to post it below as well. Thank you.